I'd like to introduce the first speaker, Bert Vogelstein now. Bert is the Clayton Professor of Oncology and Pathology and co-director of the Ludwig Institute at the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins, and is an investigator of the Howard Hughes Medical Institute. Bert was the first scientist to determine the molecular basis of a common human cancer. He and his colleagues demonstrated that colorectal tumors result from the gradual accumulation of genetic alterations in specific oncogenes and tumor suppressor genes. In 1989, Dr. Vogelstein discovered that p53 not only played a role in human tumor genesis, but that it was a common denominator of human tumors, mutated in a majority of them. More recently, his group has established that the p53 gene is more frequently mutated in cancers than any other gene. In 1991, Vogelstein and his colleagues Kinsler and Nakamura discovered another tumor suppressor gene called APC that was responsible for familial adenomatous polyposis, a syndrome associated with the development of numerous small benign tumors, some of which progress to cancer. Vogelstein and Kinsler subsequently showed that somatic mutations of APC initiate most cases of colon and rectal cancers. Dr. Vogelstein and his colleagues were also the first to map cancer genomes and to use genome-wide sequencing to identify the basis of a hereditary form of cancer. His team has determined the genetic landscape of more than a dozen types of tumors, and together with their earlier studies, this work has provided the conceptual basis for what now is commonly called personalized or precision medicine. Dr. Vogelstein has won numerous awards for his pioneering studies on the pathogenesis of human cancer. These include the Young Investigator Award from the American Federation for Clinical Research, the Bristol-Myers Squibb Award for Distinguished Achievement in Cancer Research, and the American Cancer Society Medal of Honor. He is a member of both the American Academy of Sciences and as well as the National Academy of Sciences, and of course he was one of the 11 recipients of the inaugural class of the Breakthrough Prize in Life Sciences. Bert has published more than 450 scientific papers since 1976, and according to the Institute for Scientific Information and his Wikipedia entry, his papers have been cited more than 200,000 times, and remarkably more than those of any other scientist publishing during that entire time period. So what a treat to be able to hear Bert, and I'd like to welcome him to the podium at this time.
Thank you for the very kind introduction. It is indeed an honor and a privilege to lead off what will undoubtedly be an exciting day. And can you hear me without this? Okay. So what I'd like to speak about today is cancer genomes and the war against cancer. And let me begin with when this war was started, which was a little over 40 years ago by then President Nixon, through the National Cancer Act. Now, this was a gutsy move. Imagine that the United States was the subject of a terrorist attack, but the terrorists no terrorist group claimed responsibility. What would you do? There's no enemy. You don't know who it is. How would you retaliate? And most importantly, as I will emphasize in the last part of this talk, how do you prevent such attacks from coming in the future? So back in 1971, cancer really was a mystery. We did not know who the enemy was. That has completely changed in 2013. The enemy has been identified, as I'll go through in a few minutes, and our task now is figuring out how to defeat that enemy. And this slide encompasses the work of many groups throughout the world who have revealed the genetics of the enemy in exquisite detail. Close to 4,000 tumors which have been sequenced at a genome-wide level in close to 150 studies. And each of the parentheses next to those tumors represent the average number of genetic alterations in tumors of that type. So we now know this enemy quite well, at the genetic level, and virtually every form of cancer has been studied in this detail. And we can construct so-called genetic landscapes of these tumors. Here is one genetic landscape of a pancreatic cancer patient called PA14. And it's representative of most landscapes of most common solid tumors. You can see that there are a few mountains which represent genes which are frequently mutated in this sort of tumor type. Those mountains include KRAS, mutations of which were discovered by Bob Weinberg's group, who will be speaking next, and still remains perhaps the greatest arsenal in our potential arsenal in our armamentarium, both for diagnostics and therapeutics, as I'll get to later, but also P53. The purple bars represent point mutations, and the yellow peaks represent either amplifications or deletions. If it's a tumor suppressor gene, it will be deleted; if an oncogene, amplified. Each of these white dots in the picture represent a mutation, as do the black dots in this particular tumor type, PA14. And what you can see is that some of these dots, those generally under mountains, are driver genes. I will explain that in just a second. The others, most of them, roughly 90%, are what's been called passenger genes. Now, the definition of a driver can be seen in this slide. Normally, in all adult cells, even of rapidly renewing tissues, such as the gut or the skin or the bone marrow, cell birth precisely equals cell death. If it doesn't, that's a problem. That's a disease. If there's more cell death than cell birth, the tissue will atrophy. On the other hand, if there's more cell birth than cell death in an otherwise normal environment, that really is a hallmark of the neoplastic state. That's what distinguishes normal cells from tumor cells at the physiologic level. And what all cancer genes do, whether they be oncogenes or tumor suppressor genes, is they raise that value of S, the difference between cell birth and cell death, to greater than 0. Passengers do not do that. They simply go along for the ride. Every time a cell divides, it acquires roughly 1 or 2 mutations. And if those mutations occur in a cell which subsequently acquires a mutation in a driver gene, then they will be carried along for the resultant cell expansion. How many such driver genes are there? In the most common solid tumors that account for 90% of the total cancers in the world, there are actually a limited number. By my count, only about 150. A little more than half are tumor suppressors. The others are oncogenes. So these are less than 1% of the total human genes, and these really are the enemy. In addition, these 150 or so genes have been shown to function through an even smaller number of pathways. They can be organized into about 12 pathways. The ones in blue control cell fate. They're ones which have been studied for years, ones for which the components of are Notch, Hedgehog, APC. Others control chromatin modification, epigenetics, and obviously these genes are critical for determining how cells behave, whether they become stem cells or whether they become differentiated cells, and these genes often are mutated in cancers. The second class is genes which largely control cell survival. They're those like EGFR, which allow cells that have alterations in them to grow when EGF is limited. Similarly, KRAS allows cells to grow in environments where glucose is limiting. that has obvious benefits for a cell and raises that critical selective growth advantage S to higher than 0. And then there's a small number of genes like BRCA1, BRCA2, P53, and others, which don't directly control cell growth or cell death, but rather control the rate of mutation of all genes. And when they are defective, genes accumulate more quickly and the process is accelerated. Now, this slide summarizes not just geneticists' work, but is really a tribute to work that has taken place over 20 to 30 years, and for which many of the breakthrough prizes recognize. This is the work of not only cancer biologists, but perhaps even more importantly, cell biologists, developmental biologists, and many other types of biologists who have studied the ways these pathways act, not only in human cells, but in Drosophila and C. elegans and mice. And I view this diagram that encompasses cancer research as of today really as a tribute to the worldwide effort to understand cancer that was initiated by this act in 1971. So that's where we are. What do we do with it? All of this work, obviously, has only been done because everyone who has done it hoped that it would lead to a defeat of the enemy, in this case, cancer. The first attack was done through conventional warfare. That is, standard chemotherapeutic drugs. And that has proved enormously useful in some subtypes of cancer, such as germ cell tumors or Hodgkin's. But in general, there's too much friendly fire. The host is injured, often as much as the tumor cells. So this is why we are all so excited about the idea of targeted warfare. This generation, the new generation of drugs, which will be discussed by Charles Sawyers later today, targets particularly the mutant genes that are actually responsible for the neoplastic process. And as I said, I think most of us would agree that this is the best current hope for controlling cancer therapeutically. Now, here's an example. A patient with metastatic melanoma who, as you can see, has hundreds of metastases that are distributed throughout his body, including the skin. After treatment with a BRAF inhibitor, only 4 months later, all of these metastatic lesions have disappeared, every single one of them. It is nothing short of miraculous. Unfortunately, just 2 months after that, 6 months after initiating therapy, all of the lesions have reappeared. And it's not only that they have reappeared, but if you look closely at where they have reappeared, they're at identical places. These are not new lesions. They're recurrences of the same lesions. This reoccurrence at the same place and roughly the same time within a couple month period seems almost as miraculous as their disappearance. And this is both sad and intriguing. How could it happen? Well, in a war against terrorism, war against other foes, the Pentagon and the White House has, in reality, a war game panel composed of generals and theoreticians. And our lab also has a war game analyst team, in this case, led by mathematicians, Martin Nowak, who's the head of evolutionary biology, and his colleagues at Harvard. And we set out to try and understand how these recurrences occur so commonly and so contemporaneously. And what Martin and his colleagues did is model an actual patient who has a tumor load that's hugely increased compared to what we see in experimental animals. These are literally hundreds of billions of cells. This is a typical patient who might be on a phase 1 trial, multiple metastases, each containing many tens of billions of cells. And through a rather straightforward mathematical simulation, Martin could conclude that recurrences essentially are inevitable. The only assumptions involved are that cells accumulate mutations at a rate equivalent to normal cells, and that there are a certain number of cells in a tumor which you can compute simply by measuring their diameters on CT scans. And this slide summarizes the predicted recurrences after treatment with an EGFR blockading antibody, or in fact, any targeted therapy. Now, to determine how close this prediction was to reality, Luis Diaz at Hopkins and Kelly Olner at Amgen conducted a study on exactly this kind of patient cohort, patients who had responded to EGFR blockade, that is with antibodies like cetuximab or the Amgen antibody, and then recurred. And they found that they could detect recurrence in the plasma through secreted mutant DNA molecules, which I'll describe later, at a time very similar to that predicted by Martin. And clinical recurrences came just a few weeks later. And again, this applies not only to EGFR blockade, but it applies to any targeted therapy. If you believe in evolution, which of course you all do, then you have to believe that this recurrence will occur. If you don't see it, the drug is not working in the way that we think targeted drugs should. It is a fait accompli. Now, there are ways, at least theoretically, of getting around this fait accompli, and I think Charles Sawyers will discuss some of them. But this fait accompli certainly limits the potential of targeted therapies. Now, what else can we do? Well, in a real war, if one is having difficulty, one recruits allies. And we can recruit allies too. In this case, they're the body's own defenses, the immune system. In a collaborative study with Jim Allison's group long ago, we showed that about 10% of the mutant molecules, the mutant genes in a tumor, were predicted to encode epitopes that should be recognized by the individual patient's own HLA system. And this kind of result was really exactly what cancer immunotherapists have sought for a long time. Foreign antigens, never before seen in the patient's own immune system, but now clearly there as a result of the cancer, suggesting new avenues for immunotherapy. And these potential new avenues for immunotherapy have been remarkably potentiated by Jim's pioneering work with CTLA-4, and now more recently with immune checkpoint blockades with PD-1 and other similar agents. Now, these agents, as most of you know, can induce durable remissions. And let me tell you an anecdotal story about one such patient. In the initial study published in New England Journal earlier this year with anti-PD-1 therapy, there was one of 19 colorectal cancer patients who had a dramatic response to anti-PD-1 therapy. As you all know, I'm sure this therapy works wonderfully on melanomas, some renal cell cancers, a significant proportion of lung cancers, but rarely on colorectal cancers. In this case, only one of 19. Now, having studied for colorectal cancers for decades, this result was particularly intriguing to us, and we tried to find out why. The complete remission in this patient has lasted greater than 4 years, and as I said, only 1 of 19. We suspected, or at least hypothesized, that this patient might have particularly immunogenic tumor because that patient might have had lots more mutations than the others. And we thought this patient might in fact have mismatch repair deficiency, which we tested for and found. The patient had literally 25 times as many mutations as most patients with colorectal cancer. Thousands of mutations rather than 50 mutations. And this stimulated us to initiate a clinical trial, us in this case being Luis Diaz. And I'll just show you the first patient on this trial, which was enrolled about a month ago. This is another patient, a female in this case, 56 years old, who had treatment refractory metastatic cancer and had mismatch repair deficiency, so thousands of mutations in her exome. The patient was progressing rapidly, then started with anti-PD-1 therapy and had a dramatic response to CEA, to anti-PD-1 as manifested by this graph of CEA levels. It's now even a few weeks later than when I made this slide. The CEA level is normal and the patient is back to work. Now, when I first saw these results, my first reaction was, let's stop the trial, because I've been around long enough to know we're not going to do any better than this. It can only get worse. It's anecdotal, for sure. But the point I want to make is that in the future, I think what we'll see is more of this triangle, the connection between therapeutics, genetics, and immunology. Not just genetics and small molecule therapeutics, but between genetics and immunotherapeutics. Now, in addition to these kinds of attacks on the enemy, there's one that's favored by many skilled in the art of warfare, and those are preemptive strikes. Let me show you some differences between what is practiced in cancer research and what is practiced in other types of research. This slide summarizes deaths from cancer in the US in the last 60 years, since 1950. Now, look at cardiovascular and cerebrovascular diseases. They have decreased almost exponentially by 75%. An amazing achievement in medical history. Look at cancer. It has not decreased when measured by age-specific incidence, hardly at all. Obviously, there have been successes in individual, generally rare tumor types, but to date, it hasn't made a huge impact. This is food for thought. My thought is that the reason the cardiovascular and cerebrovascular fields have gotten such prominent results is because they have concentrated on preemptive strikes. A patient who has a massive heart attack or a massive stroke is as likely to die in 2013 as he or she was in 1950. The difference is that there are less massive strokes and less numbers of massive heart attacks today than there were in 1950 because of alterations in diet, because of chemopreventative agents like Lipitor, because of early interventions like stents, and many other similar approaches to reduce advanced disease. Whereas in cancer, we as scientists and moreover the public have been entranced by this idea of curing advanced cancers. Now, I don't think that necessarily has to be the case, And I'm sanguine about the possibilities of doing prevention and early detection research for these diseases, in part because of the following results. Again, working with Martin Nowak and Sandy Markowitz, we estimate that it takes 30 years from an initiation of a colorectal cancer through an APC gene mutation to the development of widespread metastases. And the widespread metastases actually only occur in the last 3 years of this long process. Let me put that another way. There will be approximately 55,000 people who die this year from colorectal cancer in the United States alone. Almost all of them will have died only because their tumors were not detected in the first 27 years of the tumor's existence. That provides a huge window of opportunity to intervene in the process, to, in a sense, put in a stent before the massive heart attack by taking out the tumors when they're adenomas, benign tumors, or when they're early cancers before they have spread to the liver. And one of the ideas for implementing this approach is to use the genetic alterations themselves as biomarkers. A physician would carry out a test, just like physicians do every year on their patients, but they would be looking for genetic alterations in the blood or other bodily fluids. Now, these biomarker types, the genetic alterations themselves, should be distinguished from conventional biomarker types, because the conventional biomarker types are simply associated with neoplasia. They are not the proximate cause of neoplasia, as are the genetic alterations. So in our group, we have been trying to develop such strategies. One such strategy was developed by Isaac Kindy, a very talented MD PhD student, who developed a way to find rare mutations in patient samples using next-generation sequencing. This is called molecular barcoding, or a safe sequencing system. And here are some of the results that Isaac and his colleagues have obtained for 4 different relatively common tumor types. The yellow bars represent advanced cancers, metastatic cancers in the main. And as you can see, you can detect circulating tumor DNA. These are not circulating cells. This is free-floating tumor DNA, mutant DNA, in 85, 90, 95% of all these patients. Now, this may be good for following patients with the disease, for judging the responses to certain agents, including immunotherapeutic agents, but it's useless for prevention. It's useless for early detection, because once a patient has advanced disease, the horse is out of the barn. More importantly, the relatively early stage disease, not advanced. These cancers are all potentially curable by surgery alone, perhaps combined with adjuvant therapy, and the detection rate ranges from 40 to 75%, depending on the tumor type. And this general approach can be applied to any bodily fluid. One of our favorites is the Pap smear, actually, and this is the group in our lab who has done this work, Isaac and his colleagues. And the modern Pap smear, as I'm sure some of you know, is not done the way it was originally done by Papa Nicolau 60 or 70 years ago. It's done by taking a scraping from the cervix and putting it in a fluid and looking for HPV DNA, in addition to putting it on a slide. And Luis and Isaac envisioned that this same vial of cervical cells could be used not only for detection of HPV DNA, but for mutations. and in fact, for mutations that might occur in not only cervical cancer cells, but more importantly, in endometrial, uterine tumors, as well as ovarian cancers. So they designed a study based on this principle that ovarian cancers or endometrial cancers would shed mutant cells into the cervical lining, which could then be scraped and analyzed for these mutations, designed a molecular genetic test based on next-generation sequencing to detect rare mutations, and found them actually in a significant percentage of pap smears from cancer patients. These mutations were absent, of course, in all patients without cancer, but in patients with uterine cancer, they could detect them in over 90%, and in patients with ovarian cancers, not as high, but still respectable. Now, they could even detect them in very early stage ovarian cancer, stage 1. Every ovarian cancer detected by such a method is potentially a life saved. If one had a therapy that would cure 41% of ovarian cancers, there would be a ticker tape parade for that group of investigators in New York City. If you design an approach that will prevent 40% of ovarian cancers, I guarantee you there won't be much hoopla, and there are a lot of societal and economic reasons for that, but this is, I think, a viable approach, and I'm agnostic about the precise approach. It's not just genetic alterations that could allow early detection. It could be epigenomic alterations or alterations in expression or proteomics or metabolomics, as you'll hear from Luke Cantley later today, or my favorite, imaging. But I think there are a lot of opportunities. And I'll leave you with this thought. Right now, for many reasons, Plan A for getting rid of this enemy, for defeating this enemy, is the treatment of advanced cancers. Plan B, for which there's much less funding and much less research, probably 100 times less, is for preventing, either through primary prevention or through early detection, the development of these advanced cancers so they can be cured by relatively simple and straightforward surgical methods, perhaps combined with adjuvant or new targeted therapies. In my view, this should be reversed. Plan A should be preventing advanced cancers. This, of course, will not always work, so there will always be a need for new treatments. But then Plan B will be treatment of those cancers which have escaped the preventative and early diagnostic measures. I'm extremely optimistic about the course of cancer research and our ability to defeat this enemy. Medical history has shown that once one understands the disease, it's only a matter of time, sometimes a long time, but only just time before that disease is conquered. However, I believe that we will only be able to conquer this disease if we use all of our forms of warfare against it, including preemptive strikes, early detection, and other types of preventative measures. And finally, I'd like to disclose and thank our industrial partners who are trying to get some of the tests that we've developed into the clinic. Thank you very much for your attention. What do I do? Sure. Okay. I think I have time for one or two questions. I was watching the clock. I think I finished right on time. Yes. Yes, Alex.
Basically, it's about the role of immunotherapy versus targeted therapy, say with small molecules, because resistance arises non-randomly, and some of this resistance gene mutations can be predicted with significant probabilities.
Yes.
simultaneously with the initial clones. And so is the jury still out whether this approach will carry the day,
Yeah.
making immunotherapy perhaps useful but not essential, or whether immunotherapy branch of the effort will always be essential in the presence of small molecule approach?
Yeah, that's very good. It's actually a couple of questions. So, yes. So the first question is, what are the abilities of combination therapies? That relates to it. And that's a very important question. I didn't have time to go over it, but Martins, our war gamers, have shown reassuringly that if you have two targeted therapies with small drugs, you can eradicate even advanced cancers. Now, there's a catch. First of all, targeted therapies so far are all against the oncogenes. If you're going to have, you can't target a tumor suppressor gene because there's nothing to target. It's gone from the cell, it's inactivated. There are very few solid tumors which have more than one activated oncogene. In fact, most of them have 0 or 1. That's one potential problem. The second problem is that if two drugs are to work, at least theoretically, they can have no cross-resistance. If you have cross-resistance, you're very little better off with two drugs rather than one. And unfortunately, so far, the available evidence indicates that there are many mutations which can confer resistance to different classes of targeted drugs. Now, we all hope that will be overcome, and drugs with completely different mechanisms of actions for which you can have cross-resistance mutations will occur, but that remains to be seen. The second question is immunotherapy. Now, immunotherapy in combination with targeted therapies, of course, are an excellent idea. One of the concepts that's most intriguing to me about immunotherapies is that they work so well, because you can imagine numerous ways for cancers to become resistant to immunotherapies. All they have to do is lose HLA molecules on the surface or any component of the processing, or delete the mutant antigens. I mean, theoretically, you can imagine many different ways, yet they seem to work so well. This suggests to me that they're not working exactly as we currently believe they are. For example, with PD-1, there was initial evidence that it depended on PD-L1 on the surface of the cancer cells to start to stimulate the inhibition. I believe that has not been substantiated in further studies. So I think there's something incredibly fascinating going on which we don't yet have a grip on, and if we come to grips with that, we may be able to enhance immunotherapy further.